Assalamualaikum and hi guys. Today I just want to share some topic with you.
Three, two, one.
Transcription is the process of taking a single gene of DNA and copying it into a structure called mRNA. Translation is the process of taking this mRNA strand and using it to produce a protein. So first, I will talk about transcription. In this image, we can see a small section of DNA that contains two strands coiled into a helix. This is how normally found in the nucleus. But to make it easier to understand transcription, let's uncoil the two strands and show using this simplified diagram. We make example using this region here between the two lines, the gene that we want to copy. The process starts with an enzyme called RNA polymerase, which binds to the DNA just before where the gene starts. Then, just ahead of the RNA polymerase, the two strands of DNA spread apart so that all the bases are exposed. The RNA polymerase is basically going to move along DNA strand and read the bases one by one and use them to make an mRNA strand. mRNA bases will be complementary to DNA bases. So we will start the mRNA strand with complementary G base. Next, it will read T base, so it adds complementary A to the mRNA. Then, it will move to A base and add complementary U and go to G and add complementary C and so on. RNA polymerase is going to carry on doing this, pulling out the mRNA base by base as it moves along the entire gene. Once it moves along the entire gene and finish making the mRNA strand, RNA polymerase will detach from DNA. DNA strand can close back up. The mRNA is ready to leave nucleus and head to a ribosome to start translation process. To start the process, mRNA strand and ribosome bind together and start building the protein by adding one amino acid at a time. Amino acid themselves prove a ribosome by molecule called tRNA. tRNA molecule have amino acid at the top and anticodon on the bottom. Anticodon is sequence of 3 bases, which complementary to the 3 bases on mRNA that code for amino acid at the tRNA is carrying. So, because of each type of tRNA molecule is specific to a particular triplet, it ensures it always brings the correct amino acid. For example, the first triplet of mRNA sequence AGU, it will attract tRNA molecule with anticodon UCA. tRNA bring amino acid serine because AGU is code for serine. We look at second triplet CCA, it will attract this tRNA molecule with complementary anticodon GGU. tRNA carrying proline as amino acid. The code for proline is CCA. Now, the whole point of this seemingly complex process is that the tRNAs have brought the correct amino acid down to ribosome in correct order. So, ribosome is able to join them together and start whole chain of amino acid. Once it is joined, these first two amino acids together, ribosome move along the mRNA slightly. So another tRNA come down and bind its complementary codon. Bring the next amino acid. This allowed First, tRNA detach and repeat the process, but the important is it leaves amino acid behind, and the process continue until the very end. At this point, amino acid chain will detach from the ribosome and chain full up itself to form protein.
And that is all from me. Thank you.